Assalamualaikum and good day everyone. Today I am excited to share our research focusing on a critical environmental and food security issues within one of Malaysia's most valuable marine ecosystem, the Matang Mangrove Forest Reserve or MMFR. Located on the northwest coast of Peninsular Malaysia, the MMFR spans over 40,000 hectares and is globally recognized as the longest sustainably managed mangrove forest in the world. This pristine ecosystem serves as a vital natural shield against coastal erosion, a massive carbon sink, and most importantly, a premier breeding ground and sanctuary for diverse marine life. Central to this ecosystem is the Asian sea bass, Lattes calcifer, locally known as yakap. The river channel and estuaries of the MMFR host extensive cage aquaculture activities, making this fish a cornerstone of a local livelihood. To local communities relying on this Matang Mangrove Forest Reserve, Asian sea bass is a vital economic lifeline and a primary source of affordable protein. However, rising microplastic pollution in these estuarine waters raises serious food safety concerns regarding the health of this staple food. In this study, we evaluate the molecular and physical damage these microplastics inflict on the Asian sea bass and propose a biological probiotic-driven solution to protect them. To understand this threat, let us deconstruct how microplastics enter and destroy the fish physiology in 4 distinct steps. Step 1 is environmental ingestion. Highly buoyant polyethylene microplastics, fibers, and fragments circulate through the MMFR waterways. Sea bass larvae and juveniles mistake these particles for plankton, consuming them directly or inhale them during respiration, trapping them in their mouth cavities. Step 2 is gastrointestinal accumulation and physical damage. Once swallowed, these ingestible synthetic polymers accumulate in the GI tract. The sharp abrasive edges of these fragments physically wear down the inner mucosal linings, causing severe histopathological decay, including a drastic thinning of the gut wall and shriveling of the intestinal villi. Step 3 is the biochemical stress trigger. This tissue irritation, coupled with chemical leakages from the plastics, triggers intense localized stress. This leads to lipid peroxidation, which actively destroys cellular membranes within the digestive organs. Crucially, we observe a severe downregulation or genetic suppression of the fish vital antioxidant and immune-related genes, specifically superoxidant dismutase, catalase, and interleukin-1 beta. Step 4 is systemic failure. Denied their genetic defense mechanisms, the fish suffer gut microbiome dysbiosis, wiping out beneficial bacteria like lactobacillus and allowing pathogens to thrive because their intestinal villi are degraded. They can no longer absorb nutrients, resulting in high food conversion ratios, starvation, and severely stunted growth. To solve this problem before it reaches the fish, we propose a cutting-edge biotechnological solution, which is the photobiocatalytic depolymerization and consortium assimilation. This process works in 5 highly coordinated steps. First, we perform nuclear genome transformation. We codon optimize the plastic degrading aspartase gene from Ideonella zacchaensis and fuse it to a secretory signal peptide sequence. We then introduce this to the marine diatom, Phaeodactylium tricornatum. This genetic modification ensures the microalgae continuously pump the plastic degrading enzyme out across their cell membranes. Secondly, we leverage light-driven secretion because this diatom is photosynthetic. It uses natural sunlight and dissolved carbon dioxide to power this continuous high-yield enzyme secretion, requiring no expensive organic carbon feeds. Third, the microalgae naturally secrete sticky extracellular polymeric substances. This EPS matrix acts as a physical bionet, capturing and trapping suspended microplastics, holding them in direct, prolonged contact with the enzymes. Fourthly, enzymatic ester bond hydrolysis begins. The secreted enzymes initiate a nucleophilic attack on the hydrophobic surface of the trapped plastics, breaking the tough ester bond and depolymerizing the insoluble plastic into harmless water-soluble monomers. And finally, we achieve synergistic metabolic assimilation. Operating in a mixed biofilm with native heterophytic bacteria, the algae provide oxygen while the bacteria rapidly absorbs the soluble plastic byproducts, processing them through their native TCA cycle to fully convert the plastic into harmless carbon dioxide, water, and natural biomass. While highly effective, deploying genetically engineered microorganisms directly into the sensitive MMFR estuary introduces serious ecological trade-offs that we must critically address. Our first concern is genetic escape and horizontal gene transfer. If these engineered diatoms escape containment, they risk transferring synthetic genes to wild algal or native bacterial strains, which could alter the delicate baseline nutrient dynamics of this estuary. Second is ecological competitiveness. If the engineered algae outcompete native phytoplankton in plastic-heavy waters, they could trigger artificial algal bloom. These blooms would deplete dissolved oxygen, causing hypoxia and asphyxiating the very wild fish we are trying to protect. To mitigate this risk, we advocate for synthetic biocontainment circuits. By engineering synthetic anisotropy, making the diatom depend on a specific laboratory-supplied nutrient that does not naturally occur in the mangrove, or by incorporating a genetic kill switch, any cells drifting beyond our designated treatment area will automatically undergo programmed cell death. This prevents warp establishment and limits genetic dispersion. In conclusion, while gutting fish before cooking reduces the physical ingestion of larger plastics, the threat of nanoplastics and chemical leaching into the edible fillets of Asian sea bass remains a pressing food safety concern. Bioremediation at the estuarine level is essential to secure this food source. Personally, this research has expanded my understanding of estuarine toxicology. In this research, I also responsibly used artificial intelligence exclusively as a collaborative tool to brainstorm key concepts and sketch rough drafts for my layouts. This AI assistance was particularly helpful in structuring the initial visualization framework, such as mapping out the biological pathways for the tree map and flow maps. Crucially, the final analysis, selection of peer-reviewed data, and overall research integrity remain entirely my own independent work. By safely deploying biocontained biotechnology, we can preserve the ecological integrity of the Mata Mangrove Forest Reserve, support our coastal communities, and safeguard our food supply. Thank you and have a nice day.